Much like a factory, transcription and translation are the processes by which a cell produces materials used in its daily functioning. It begins with raw materials in the form of amino acids and, using genetic information copied from DNA to RNA, assembles those amino acids according to the genetic instructions to form the finished product in the form of a polypeptide or a protein. These proteins are later packaged, similar to factory-made products, before being sent out via post-translational modification and are then sent out to their final destination. The first part of the process is transcription, which involves the copying of genetic information from DNA to RNA. This process produces the instructions for protein synthesis and can be compared to a set of blueprints for a factory. Translation involves the copying of a DNA strand into mRNA, which contains a specific genetic message later used in creating a protein. Once it has exited the nucleus and is in the cytosol, or within the rough endoplasmic reticulum in eukaryotes, the mRNA then undergoes the process of translation. Translation is similar to the assembly line of a factory, where amino acids are put together according to the RNA instructions to make a finished product, the protein or polypeptide chain. Translation begins with the initiation phase, which occurs when the smaller subunit of the ribosome and an initiator tRNA molecule both bind to a specific portion of the mRNA strand. In eukaryotes, the small subunit begins by binding to the 5' cap of the mRNA and moving down the molecule in the 3' direction until it has reached the start codon. Because of this action, the start codon initiates the start of translation and dictates the codon reading frame for the mRNA. Once this occurs, the larger ribosomal subunit then attaches to the mRNA, forming the translation initiation complex, which is brought together by proteins referred to as initiation factors. After the initiation phase of translation, the elongation stage begins, in which amino acids are then added one by one to the previous amino acid at the C-terminus of the growing chain. The elongation stage begins with codon recognition, in which the anticodon of a corresponding amino acid tRNA base pairs with the complementary mRNA codon in the A site. This process is much like the conveyor belt of a factory, as once one tRNA exits the ribosome, another tRNA enters, making the process continuous as the finished product is produced. In order for the chain to be completed, the termination phase has to begin when a stop codon reaches the A site of the ribosome. Once a stop codon is received, a release factor or a protein shaped like an amino acid tRNA binds to the stop codon on the A site and causes the addition of a water molecule instead of an amino acid to the polypeptide chain. This reaction breaks the bond between the polypeptide chain and the rRNA at the P site, releasing the polypeptide through the exit tunnel of the ribosome. After the polypeptide chain is released from the ribosome, it then undergoes a process called protein folding, in which it begins to coil and fold into a three-dimensional shape based on the properties of its amino acids. In many cases, a chaperone protein called chaperonin helps the polypeptide to fold correctly. In this stage, post-translational modification can also occur, in which sugars, lipids, or phosphate groups can be attached to make the protein functional. Some errors which occur during protein synthesis include premature termination, genetic mutations caused by insertions or deletions, while errors during splicing include exon skipping and failure to remove intron. Errors during transcription include nucleotide incorporation and polymerase slippage, and errors in folding consist of kinetic trapping and spontaneous unfolding.